Imagine trying to break into a locked vault, but the vault is microscopic and covered in a shifting thicket of thorny spikes. That is essentially what the human immune system faces when it encounters HIV. Our bodies do have the machinery to fight back. We can produce special defenders, broadly neutralizing antibodies, or super antibodies, that are uniquely shaped to find and pierce through that armor. The problem is that the virus covers its most vulnerable spots with a thick, interlocking layer of sugars. It is a literal sugar shield. When standard antibodies try to latch on, they just bounce right off. Because those weak spots are so well hidden, the body rarely gets the chance to learn how to craft the right super antibody. When it does, the process usually takes years of exhausting trial and error. For decades, the central challenge of an HIV vaccine has been speeding up that timeline. We need to teach the immune system to mass-produce these rare, shield-piercing defenders on demand. Before any vaccine reaches humans, scientists test it in an animal model, typically macaques. This is the crucial proving ground, where we see if our ideas actually work in a living immune system. But the researchers continually hit a massive wall. When they introduced standard HIV-like viruses into macaques, the animals' immune systems couldn't get past that dense sugar shield either. Because the animals couldn't consistently produce the super antibodies, researchers had nothing to observe. You cannot study a process that never happens. Without a reliable biological training ground to watch exactly how these specialized defenders are built, the search for a workable human vaccine stalled. To bypass this problem, a team of researchers engineered a custom monkey virus, creating a decoy known as SHIV.5-MUT. This decoy virus looked normal, except researchers stripped protective sugars from a specific patch called the V1 loop. This thinned-out spot acted as a biological trap. Recognizing the obvious weak point, standard antibodies swarmed and latched directly onto it. This intense, highly localized attack trapped the decoy virus. To survive the onslaught, the virus was forced to quickly adapt and mutate its outer shell. To escape the swarm of attacking antibodies, the virus mutated by physically shrinking its V1 loop even further, pulling its armor back so the attackers had nothing left to grab onto. This diagram illustrates the result. By shrinking its armor, the virus accidentally exposed a deeply hidden weak spot, the blue V3 glycan target. Now fully visible, the immune system pumped out specialized super antibodies, the green shapes, designed to lock onto that exact spot. Within a year, 14 out of 22 macaques given this decoy successfully developed the elusive super antibodies. In contrast, a control group given the unmodified virus saw 0 out of 14 macaques develop those same defenders. By forcing the virus to peel back its own armor, scientists proved that the immune system will reliably learn how to destroy it as long as it gets a clear look at the target. So, how does manipulating monkey viruses translate into a life-saving tool for humans? To be clear, researchers have no intention of infecting humans with a live, mutating decoy virus. Here, we have a detailed 3D model of the virus's surface. By closely monitoring the macaques, scientists now possess a precise molecular map of exactly what the virus's fully exposed weak spot looks like, highlighted right here in dark blue. That structural map is what we have been searching for. It outlines the exact shape a human vaccine needs to target. With that map, researchers can build a completely harmless synthetic vaccine particle. We can engineer it so it mimics that pre-shrunk, fully exposed state. This means we skipped the complicated two-step dance entirely. We can hand the human immune system the final blueprint from day one. By tricking the virus into shedding its greatest defense mechanism, scientists forced HIV to reveal its deepest vulnerability, giving us a clear, actionable path toward a human vaccine.